Alright, let's go ahead and get started with the reproductive system lab. In your lab manual, this goes from page 67 to page 70, and so after that it goes into some different structures in the back of the manual that you can utilize as different models for study material throughout the semester. Hopefully you're using some of those, but we'll go ahead and get started with objective 1 here. This is starting off with identifying organs of the male reproductive system. So I have everything on here that you'll need to be able to identify from this point of view. And again, there is a tab in your Blackboard page that will refer you to a worksheet that you can utilize to label some stuff while we're going through this at the same time if you want, or do it maybe afterward for an extra study. Otherwise, there are models that you need to be able to identify these different structures on. So not only do you want to be able to identify them from these points of view, but also from the models, because you will see those models on the quizzes, just like usual. All right. So starting off, we have our testes down here. So this would be the teste, and then surrounding the teste would be the epididymis. All right. And then these are held within the scrotum. So you can kind of see this sac-like structure going around it is the scrotum. And then stemming from the epididymis, we have the vas deferens, also listed as the ductus deferens, or the spermatic cord. All right. So the spermatic cord is kind of the initial structure that comes off of it, and the vas deferens is what, or the ductus deferens is kind of what's coming off of it. So you can put either of those and you'll get the correct answer. It might mark it wrong initially, but I will manually go in and make sure that both answers are accepted. And then we end up moving through the male reproductive system here, and we start passing through some different structures. So the first structure we pass through that we need to be able to identify is called the ejaculatory duct. All right. And then along the way, before we get to this actual point, this actual tube point, the seminal vesicle leaks into that ejaculatory duct. All right. So the seminal vesicle is kind of behind the vas deferens at this point, and the point at which it joins with that is called the ejaculatory duct. All right. And then we continue onward, and you can kind of notice as we're passing through this little spot, there's this red structure behind us here, and this is a gland known as the prostate gland. All right, commonly cancerous in older gentlemen. So this is something that you'll have to get checked on if you're a man when you start getting to a little bit later stages in life. And then after the prostate gland, we're still moving along in the vas deferens, and we start getting another tubular structure going down here. But before we get there, from this little region here, we have our prostatic urethra. All right. So basically, this is going to be coming in from the bladder region. So this is a urethra that's passing over top the prostate, and then it joins with the vas deferens. And so that's our prostatic urethra. So it's just named the prostatic urethra because it's by the prostate, and it's still a urethra because it's going to be what is containing the stuff that's going to be coming out of the penis, basically, either the urine or the sperm at the time. And then once we have passed our prostate, now we are in the next part of the urethra called the membranous urethra. So now we're past the prostate and we have made it to our membranous urethra. And we just keep going, keep going, keep going. Now we're in the penis. So this structure is known as the penis, and so we're going to call that part of the urethra the penile urethra. And so those are all your different regions along the vas deferens that you need to be able to identify. All right, spermatic cord, vas deferens, your ejaculatory duct, your prostatic urethra, and then your membranous urethra, and then your penile urethra. All right, and then there's a couple structures along the way that you'll want to be able to identify, like the seminal vesicle and the prostate gland. And then we also have this bulbourethral gland here. Also, you could put Cowper's gland along the way that kind of leaks into the membranous urethra. All right, so helping supply that sperm with different things that are going to help nourish it or make it motile. We learn that kind of stuff in lecture. So those are the different structures associated with Objective 1. And moving along with Objective 2, we can start looking at some stuff such as like the corpus cavernosum here, which is going to be a more erect part of the penis. It's going to have some more erectile aspects to it. We have the foreskin right here, which is going to serve as a protective function but leads to a lot of infections if it's not cleaned properly. So in a lot of cultures, they just remove it. We have the glans penis, which is the actual tip, if you will, of the penis. And so we have our glans penis here. And then you have the corpus spongiosum. All right, so the corpus spongiosum would be kind of this little region that's going along behind the corpus cavernosum. All right. And next we can identify objective 3, some spermatozoan aspects. And so a sperm is basically going to be, your little flagella, your tail. You could say either and get credit for it. The mid piece, which is kind of the middle of the body. This is where a lot of the power of powering that tail is going to occur because of the mitochondria there. And then we have the head, which is going to contain the nucleus and DNA. And then the actual tip of it is called the acrosome. And this is how it penetrates into the egg, into the protective layers of the egg, with digestive enzymes. So these 4 different regions you should be able to identify, as well as if I ask what would be contained in the head, and you would tell me a nucleus. Moving on with objective 4, we need to be able to identify a couple different things histologically. And so what you're looking at here is a cross-section of some seminiferous tubules inside of the testes. So these seminiferous tubules are going to be consistent of spermatozoans, spermatocytes and spermatids. And so the outside is going to be where all the spermatocytes are, and then they differentiate and grow inwardly until they reach the inside of the tube where they become spermatids, and then they leave through the tube and then out into the epididymis and where they're stored and then eventually ejaculated. All right, so this inside is where the spermatids would be. So if I point to the inside and say, what would be found here? You would tell me spermatid. And I point to the outside and say, what cells would be found here? You would tell me spermatocytes. You don't need to tell me primary, secondary, or anything like that. Just that it's where you would find spermatozoa. All right. And then also you need to be able to identify that these are interstitial cells in between all the seminiferous tubules. So all the big circles are seminiferous tubules. Everything in between would be the late egg or interstitial cells, helping with testosterone. All right. So these would be the interstitial cells. When it comes to the ovary, we're going to have 2 different views that you should be able to identify: primary and secondary. With the primary, it's going to look really small like this, and you're going to have a circle, and this would be considered the primary oocyte, the primary egg, and then B would be the primary follicle, supplying that egg and keeping it alive. When it gets really large and ready to be ovulated, this little egg is still the same. Everything surrounding that is the follicle. So this is our secondary oocyte, and then this would all be the mature follicle that's surrounding it, keeping it alive. When it comes to objective 5, we're looking at some more parts of the reproductive system, but it's just female related. And so you need to be able to identify that this is an ovary right here. This is a cross section. So we're looking at an ovary here. And then stemming from that ovary, we have these fimbriae that are going to catch the egg, the newly discharged egg. These fimbriae are going to help guide it in like big fingers into the infundibulum of the uterine tube. All right, so this little spot here would be called the infundibulum, where the egg first enters. The fimbriae guide it into there, and now we're in the uterine tube. And then or you could say fallopian tube too, same thing. And then from the fallopian tube, then we enter into the uterus. And so the uterus has 3 different aspects that you should be able to identify. First, you want to be able to identify this is the uterus. Second, the first region it enters is the fundus. The middle part is the body, just like when we looked at the stomach. And then the end part is the cervix. All right, this is where baby's head is going to push to stimulate oxytocin release and more contractions via that positive feedback loop. Sorry. So you got your fundus, body, and cervix of the uteral organ. And you have different linings of the uterus that you should be able to identify. You have your endometrium, which is the inside lining. If you have a diagnosis of endometriosis, it's people that typically have an issue with getting pregnant because this endometrial lining is so thick. And this is also the lining that is shedding when we're talking about the female reproductive cycle. And then the meat of the uterus is the myometrium, muscle. So a lot of the contractions occur in the myometrium. And then the perimetrium is kind of this outside layer. So peri is surrounding, so it's surrounding the organ. Myo is muscle, it's the muscle of the organ. Endo is the inner lining. So you can kind of just piece stuff together by their Latin roots. And then we have our vagina here, which is just this vaginal canal that leads out of the reproductive system of the female. Now a little bit different view for some different structures we should be able to identify. Again, you have your ovary here. You would have your fimbriae kind of ready to guide that egg into the fallopian or uterine tube here, into the uterus, and you just need to be able to identify that this is the uterus. You might want to be able to identify that this is the cervix as well right here. And then we leave out through the vagina. And you need to be able to differentiate the vaginal opening right here versus the urethral opening. It's pretty easy because the vaginal opening is significantly larger so that a penis can be inserted, whereas a urethral lining doesn't need to be large because urine just needs to be able to make its way out in a liquid, all right, from our bladder here. And so we have our urethral opening here. And then you have a couple different structures that we'll have another view here shortly, but you have your labia minora, which is kind of this minor or closer to the body labia, this little fold, and this labia majora, which is a larger fold that is more external than the labia minora. So here's another picture of that. This is looking at it from, an outside point of view rather than a cross section. You have your mons pubis up here, which would kind of be just like the very top part of the vagina. It's really not technically part of the vagina, but it's like kind of like that top part. So if we kind of point outside the vagina at this point, just on top, that's the mons pubis. And if you pull the labia minora and majora to the side, you can find the clitoris toward the top. And then you have this labia minora, which is these smaller folds, and labia majora here pulled to the side. And so these would normally be kind of crest over a little bit more. These are just protective. You don't worry about perineum, that's not on our list, or the anus. These are just for relativity aspects. And then our last objective is looking at the breast. And so this is a female breast, not a male breast. And what we're looking at is a breast right here, and then this is an areola that surrounds the nipple. So the small tip is the nipple and the outside is the areola. And then if we look at a nice cross section of that, we can find the different lactiferous glands or lactiferous ducts. So stemming right into the nipple are the ducts, so that lead the milk out of the system. So the lactiferous ducts, and where the milk is kind of produced are in these alveoli. In your objective list, it says alveolar glands. So either are acceptable. And so these alveolar glands are going to be producing that milk, and you can kind of see the flow of that through the lactiferous sinuses here, then ducts, and then milk flow out of the nipple. But that actually does it for our lab, and this is our last lab. So if you have any questions, feel free to email as usual. Otherwise, we'll chat soon.